Hey everyone, and welcome to this explainer. Okay, we're just going to dive right into this today because we are talking about something so incredibly fundamental, it is literally the basis of who you are. Think about it for a second. We might occasionally forget our own home addresses, or we might absolutely forget our ATM PIN codes from time to time. And hey, we've all been there, right? But as students of biology, there is one absolute fact that we can never, ever forget, and that is the cell is the structural and functional unit of life, period. So you might be wondering, why is the cell so unforgettable? Well, I want you to imagine a magnificent, sprawling house. That house might be massive, it might be made of thousands of different materials, glass, wood, metal, but its fundamental structural unit, its most basic building block, is just a single brick. Without that brick, there is no house, nothing gets built. In the exact same way, whether you're looking at a tiny, microscopic bacterium or a ridiculously complex human being made of trillions of interconnected parts, the fundamental brick of it all is the cell. It's the absolute smallest thing that can independently be called living. And honestly, once you truly wrap your head around the cell, all of biology just clicks. It becomes life jingalala. It all makes perfect sense. So here is our quick itinerary for today's journey into the cellular city. First, the foundation of life. Second, simple versus complex cells. Third, the protective borders. Fourth, plant versus animal cells. And finally, number 5, the cellular city organelles. All right, let's jump straight into section 1, the foundation of life. Let's start by laying down that foundation. Every single living thing relies entirely on cells to function. And this is actually exactly why a virus isn't technically considered a living organism on its own. It lacks a complete cellular structure. To be alive, to truly exist independently in the world, you need a cell. Now, it actually took us a surprisingly long time to figure this out. It started way back in 1665 when Robert Hooke first observed dead oak cork cells, which honestly were basically just the empty outer walls. Nothing was inside them anymore. Fast forward to 1831, and Robert Brown discovers the nucleus, essentially the brain of the cell. Then, a few years later, in 1838 and 1839, two scientists, Schleiden and Schwann, had this massive breakthrough. They realized that all plants and animals are made of cells, forming the basic cell theory. But there was a glaring missing puzzle piece here. Where do these cells actually come from? Do they just magically appear? Well, in 1855, Rudolf Virchow dropped a total game changer. He stated, omni cellula e cellula. It's a great phrase, and it simply means new cells only arise from pre-existing cells. You can't just spawn a living cell out of thin air. Moving right along to section 2, simple versus complex cells. If we're looking at the architecture of life, we basically have two main types of blueprints. Think of it as the simple village and the complex metropolis. We call these prokaryotic and eukaryotic cells. Prokaryotic cells, like bacteria, are your simple villages. They are incredibly tiny, like 1 to 2 micrometers. They don't have a true nucleus, so their DNA is just kind of hanging out there naked in the center of the cell. But here's the thing. Because they are so small and they don't have all these complex internal compartments, they are astonishingly efficient at reproducing. An E. coli bacterium can divide and double its entire population in just 20 minutes. 20 minutes! Now, on the other hand, eukaryotic cells, like the ones inside you and me, are the complex metropolises. They are roughly 10 times larger, ranging from 10 to 20 micrometers. They have a highly protected, secure membrane-bound nucleus and tons of dedicated internal compartments for different jobs. But because of this massive scale and complexity, a typical human cell takes about 24 hours to divide. So you see, it's the ultimate evolutionary trade-off between lightning-fast speed and intricate complexity. But of course, nature absolutely loves throwing us an exception. Meet mycoplasma, which is also known as PPLO. It is literally the absolute smallest living organism on Earth, measuring a minuscule 0.1 to 0.3 micrometers. But here's the really wild part about it. It completely lacks a rigid cell wall. Because it has no hard outer shell to keep its structure, it can constantly change its shape. It's actually known as the pleomorphic joker of the cellular world. Think about it like a joker in a deck of cards or an entertainer constantly changing costumes and expressions to trick you. Mycoplasma constantly changes its physical form to adapt and survive its environment. It's completely wild. All right, section 3, the protective borders. So if the cell is a functioning city, how does it defend itself from the harsh, chaotic outside world? Well, it uses a specialized envelope, and this is especially true in the bacterial world. Let's break down this bacterial border from the outside coming in. First up, you hit the glycocalyx. This is an outermost carbohydrate coat. Sometimes it's this loose, sticky slime layer, and other times it's a really tough defensive capsule. Step past that and you hit the cell wall. Now, I want you to think of the cell wall exactly like ambuja cement. It is incredibly rigid, totally unyielding, and its entire job is to ensure the cell doesn't burst open or collapse in on itself under environmental pressure. And just as a quick aside, scientists actually use a special dye on this envelope. If the cell wall holds onto the dye super tightly, we call the bacteria gram-positive. If the dye just washes away, it's gram-negative. Simple as that. Finally, moving past that cement wall, the innermost border is the cell membrane. This acts as the highly selective security gate for the city, deciding exactly what gets in and what gets kicked out. Now, how that specific security gate is structured completely shifted our entire understanding of biology. Back in 1972, Singer and Nicholson proposed the fluid mosaic model. To understand this, I want you to visualize a fluid, moving sea made entirely of lipids or fats. It's just flowing. And floating within this oily, shifting sea are these giant protein icebergs. The membrane isn't a solid static brick wall like scientists used to think. It's a flexible, flowing liquid that actually allows the cell to grow, to bend, and to precisely control what enters and exits. It's constantly in motion. If we look at a really specific example, like the human red blood cell membrane, we can see it's mathematically precise. It's about 52% protein, 40% lipid, and a small 8% mix of carbohydrates and cholesterol. And I really want to highlight that cholesterol part. You know when you sit down at a restaurant and your table has that one incredibly wobbly leg? So you fold up a napkin or a piece of paper and wedge it tightly underneath to stabilize the whole thing? That is exactly what cholesterol does in your cell membrane. It wedges itself right between those fluid lipids to stabilize the whole fluid structure so the membrane doesn't just fall apart and turn to mush. It's an absolutely brilliant piece of biological engineering. Section 4. Plant versus animal cells. Stepping back into the complex world of eukaryotes for a second, we obviously know that plants and animals are very different. But if you were looking through a microscope right now, how would you quickly tell their cells apart? I love using the super simple memory trick, the 4 C's. Okay, so plant cells have a rigid sea wall for structure. They have a massive central vacuole to store water, which, by the way, is so huge it literally pushes all the other organelles right up against the edges of the cell. And they have chloroplasts to make their own food from sunlight. What about animal cells? Well, we lack the cell wall entirely, we definitely lack the giant vacuole, and we completely lack chloroplasts, which is honestly a bit of a shame because it's the exact reason why we can't just stand outside in the sun and photosynthesize our lunch. But animal cells do have the 4th C, centrioles. These act almost like biological fishing reels. They cast out tiny lines to physically pull DNA apart when our cells divide. So just remember those 4 C's and you will never ever confuse a plant and animal cell again. Which brings us to Section 5. The cellular city organelles. Let's dive straight into the cytoplasm. This is the watery arena inside the cell where all the magic actually happens. Within this bustling city, there's a highly coordinated network called the endomembrane system. It basically consists of 4 main internal factories: the endoplasmic reticulum, the Golgi complex, lysosomes, and vacuoles. Now, why are these 4 specific organelles grouped together like this? Because they act exactly like an assembly line in a giant, highly efficient manufacturing plant. The product moves sequentially from one station directly to the next without missing a single beat. First up on that assembly line, you have the endoplasmic reticulum, or the ER. The rough ER is studded with thousands of tiny ribosomes, which makes it look all bumpy under a microscope. Its entire job is to aggressively manufacture proteins. Right next to it is the smooth ER. This one lacks ribosomes, so it's smooth, and it focuses on synthesizing lipids and fats. Once the proteins and lipids are manufactured, they're shipped directly to the Golgi complex. Now, I want you to think of the Golgi as the cellular post office. It takes the newly made products, packages them up neatly, and physically attaches a carbohydrate address tag, a vital biological process called glycosylation. Why is this so crucial? Because if a package gets lost inside the cell, vital functions shut down. The Golgi ensures the cell knows exactly where to deliver these crucial building blocks every single time. But you know, every bustling city produces waste, and every city needs ample storage. That brings us to the real extremes of the cell city. First, the lysosomes. These are your dangerous waste disposal units. They are quite literally nicknamed suicidal bags because they are packed with highly acidic, incredibly destructive enzymes that dissolve cellular garbage. They have to be kept strictly contained. If a lysosome accidentally bursts, it will brutally digest the cell itself. On the complete flip side of that chaos, we have vacuoles. These are the giant, peaceful storage tanks holding water, nutrients, and yes, some waste. As we mentioned just a bit ago, in a plant cell, a single central vacuole can be so massive that it takes up 90% of the entire cell's volume. Just huge pools of storage. Now, we mentioned ribosomes a minute ago, the little protein factories on the rough ER. But if you've ever studied them, I know exactly what you're probably thinking right now. The math seems completely, totally broken. Let's look at it. Prokaryotes have 70S ribosomes, which are made of a 50S and a 30S subunit. Eukaryotes have 80S ribosomes made of 60S and 40S subunits. Wait a minute. 50 plus 30 equals 80, not 70. And 60 plus 40 is 100. It doesn't make any sense, right? So the absolutely crucial point here is understanding that the S stands for Svedberg unit. It measures density and how things settle in a centrifuge. It does not measure straightforward weight. Think of it like this. Imagine taking a very heavy, dense bowling ball and then throwing a light, fluffy blanket over it. The total weight of the object obviously increases, but the average density of the combined object actually drops. That is exactly why the combined subunits don't mathematically add up like standard numbers. They change their aerodynamic density when they lock together. It's literally physics at a microscopic scale. Honestly, isn't that just absolutely incredible? Every single time you peel back a layer of this topic, you just find more breathtaking engineering perfection. From the fluid, stabilizing lipid borders that are guarding the perimeter, to the highly coordinated post office tagging of the Golgi complex inside, all the way down to those density-driven protein factories reading your DNA, the cell is truly a miniature, bustling metropolis. It is executing every essential function of life flawlessly, all within a tiny boundary that you can't even see with the naked eye. So, I want to leave you with this final thought today. We've just spent this entire explainer marveling at how a single, solitary, microscopic bacterium can independently survive, divide, defend itself, and perform all the vastly complex functions of life. It is a completely independent powerhouse. Now, I want you to look down at your own hands for a second. You are not made of one cell. You are a cooperating universe of trillions of these incredibly complex metropolises, all working together in perfect harmony, just to make you you. If one single cell has that much immense power, what untapped potential exists within the trillions of cells that make up your body? Think about that. Thank you so much for joining me on this explainer. Keep learning, stay curious, and I'll catch you next time.